Good morning, everyone, and apologies for the technical glitches. This is this is going to be, I think, more of a workshop than a conference, and people will be joining on later in the day. So I may repeat myself at one point because there's some logistical things to go through. Can they see me? Yes, okay. So I wanted to first of all say, so I'm Vincent Nguyen, I'm the co-director of the Global Health Center, I'm a professor of anthropology and sociology here, and I'm a physician and also the PI of the FINPHARM project, which really has been piloted, as we say in French, by Nadia here and earlier Ryan Whitaker, who's now at UCSF. And I'm going to speak a little bit about the kind of genesis and evolution of this project because I think the history is important. But before I go into those details, and I'll speak for about maybe 10-15 minutes, I won't speak much more. I should say, first of all, the conference is being recorded and will be made public on our YouTube channel and website in the coming days. If you do not wish to be on this recording, let us know after the conference or send your request to globalhealth@graduateinstitute.ch. For those of you who are online, please type your questions or comments in the chat box by selecting everyone, including any addressed directly to an individual speaker. So if you want to send me a question, send a message to everyone. That way, when I answer, people will know what I'm answering. Also, perhaps most importantly for us, we would like to invite you to register your email with us. European privacy rules mean that we cannot harvest your email, so if you don't do this, you will disappear from our radar screen. That will allow us to follow up with a shared bibliography, news reports, upcoming publications, and forming a community of practice around antibiotic crises. We will not be spamming you. So you can scan the QR code on the screen or click on the link dropped in the chat box if you're interested. So I've covered all the logistical issues for now, and I may have to come back to those at the beginning of the next panel. And what I wanted to do now is give you a little bit of background, and this is going to be the more anthropological stuff. So we're going to be evolving over the day towards perhaps more practical stuff, practical and closer to real world things. But I wanted to explain the genesis of the FINPHARM project, which is about financialization in the pharmaceutical industry. The starting point for this project was an earlier ERC, which was on HIV and the advent of antiretrovirals and treatment as prevention and a range of other developments in the field of HIV where we found, became pretty clear that antiretrovirals were being repurposed from individual clinical indications towards more population-based indications. And one of the things that was interesting in that was the role of how this was shifting the way pharmaceutical companies were investing in antiretrovirals. So the initial premise of this project was that pharmaceuticals were a kind of a unique and mysterious commodity in the sense that they have, unlike other types of commodities, their value lies very much in the future, and it lies in calculations made about their clinical or population benefit. So that, I think, makes sense to everyone. I'll give an example. I take two medications, sorry to get all personal. I take two medications for high blood pressure. The value of those medications for me is that they reduce my statistical risk of having a stroke or an MI, like many, many other people. The value to the pharmaceutical industry is that there's a market of people who have blood pressures over a certain amount, 140/90, and that they will be able to sell the medication to treat those people. The value, but there's a value also to the payer, which is an insurance company usually, or a national insurance scheme, which is decreased medical costs. And what we've seen in this project is that these three kinds of values, and there may be others, play a delicate dance in the way that people decide whether or not to invest in something or develop a drug. There's individual clinical there's clinical benefit, there's population benefit, and there's return on investment in terms of a market. Now, all of these are subject to calculations. It's not like the value of this bottle is that I drink it and my thirst is quenched, and the market is there's so many people buying bottles of water. It's much more mysterious and calculated, and it lies in the future. It's not immediate value for the most part. So what initially interested us was the parallels between these forms of calculation of future misfortune and bringing the value of something, valuing something that was a hypothetical future outcome in the present. This is, in a sense, what finance does, because finance involves buying and selling things based on the calculation of what their future value will be, assets, for example, or derivatives. And all of this, so there's a finance buys and sells things that based on calculations of future value. Pharmaceuticals are bought and sold on the basis of calculations about their future value. All these calculations are complex and are riddled with assumptions and hypotheses that we could call cultural. And that has a lot of parallels with witchcraft and divination, which are also cultural practices where future events have value in the current moment. And that was essentially the genesis of the project. And we were interested in three different spaces of value. We didn't use that term, but we could call them that way. Places where pharmaceuticals operated, the different aspects of future value of pharmaceuticals could be looked at. But before I go into those three spaces, I should also say that I'll do that afterwards. This makes more sense. So the first one was antiretrovirals and the way in which, as I mentioned before, antiretrovirals were being repurposed in different ways, using different modes of calculation of their future value. So that was a bit of a complicated sentence. Basically, antiretrovirals, once you've treated everyone who has HIV, then you saturated your population. And this is true for any treatment, blood pressure, cholesterol. So then, and this is not me saying it, this has been broadly established. So then what does the pharmaceutical industry do is it widens, it has an interest to increase the market by lowering the clinical threshold for treatment. So for example, maybe the blood pressure should be treated not at 140/90, but at 130/85. Normal blood pressure is 120 over 80. So you decrease the threshold of treatment. This happened with cholesterol medications. And the pharmaceutical industry showed that for cholesterol medications, the lower the cholesterol, the lower the risk of heart disease. Now there's a catch to this, which is that, and epidemiologists, clinical epidemiologists know this number, and I'll try to explain this very simply. Let's say my blood pressure or my cholesterol is, we'll just invent a number, 10, and I have it's an imaginary number, just for sake of clarity. And let's say at 10, I have a 10% chance of dying in the next 10 years, 10. So if you treat me, you need to treat 10 people, and you will prevent one death over the next 10 years, or one heart attack, we'll say. Let's say you reduce it to 5, and then my risk is, let's say, 3. Sorry, let's say 10%. Sorry, if you treat 10 people, you'll prevent 1 heart attack. There's a 10% risk. If it's 10, 10, 3, if you treat 10 people, you will treat 10 people, 10%, 1, 10 people, you'll prevent 0.3, and so on and so forth. So the number you need to treat to prevent an event increases because the risk is lower. The risk reduction is lower. Not sure is that clear? Yeah. So what was happening with cholesterol is that we were treating increasingly large numbers of people to prevent increasingly less events as the threshold went down. This has public health implications. Also has clinical implications because drugs have side effects and toxicities. Even if they're very rare, if you treat a lot of people, they will become, 1 in 1,000, you treat 1,000, then one person will get so you're getting a sense of this numbers game, right? That's going on with these changing of thresholds to develop new markets for medications. And that's what was already going on with cholesterol. We saw it we know this is going on with psychiatric medications where clinical trial data is being parsed or analyzed to reverse engineer diseases. Let me explain. You do a clinical trial, you give 1,000 people an antidepressant, 1,000 people placebo, great results. Of the 1,000 people who are depressed, 700 feel better, great. You've got your market for people who are depressed. But in the clinical trial, there's these very detailed questionnaires, and you notice that, people are also less anxious, or they're less we'll just say less anxious. So then you can reverse engineer a disorder called anxiety disorder, which then becomes another market. So you're basically reverse engineering the diagnoses to create new markets for medications. All this is known and in the industry, by clinicians, and in the anthropological literature. What was interesting for us was, how is this being done, right? What are the processes by which that future value is calculated? And of course, it's very hard to get at that, but it seemed to us that finance and tracking what was happening in financial markets could give us a sense of what was going on there. Because we don't have access to that thinking or that rationality. So the first space we were looking at was antiretrovirals, and what was interesting there is that a new market was being created by exploring how antiretrovirals not only could be used to treat, but also to prevent, right? So far, all of the medications that I've been talking about, the blood pressure medications, the cholesterol medications, those are just preventive, so you change the threshold to decrease future disease. Psychiatric medications, you create new diseases to be able to treat them. And here we have the creation of a new category of people who require prevention. Because who needs antiretrovirals for prevention? Well, it turns out it's high-risk gay men. And it was very interesting to see the reverse engineering of that category of people. So we looked at the antiretroviral space, and there's a lot of how should I put it? A lot has happened in that space, and we saw very clearly that there was, as I said, reverse engineering of populations to treat. We looked at also, interestingly, the Ebola space, and there, running out of time, so I'm going to sum up. There we found one very interesting thing, which is not widely known, which is that most of the platforms, so there's no market for Ebola vaccines. There are too few epidemics, but people wanted to develop an Ebola vaccine. It was funded by the US military, amongst others, for obvious national security reasons. And the platforms that were developed for these Ebola vaccines, most of them then became COVID vaccines. So there was an interesting finding there about how national security produced a public investment, which then pays off subsequently for other vaccines. And finally, we'll turn now to what today is about, is the antibiotic resistance space. And I wanted to just say three things about antimicrobial or antibiotic resistance. So a bit of a important qualification here. Antibiotics are drugs. Antimicrobials are things like bleach, heavy metals. Soap is an antimicrobial, disrupts bacterial walls. So when we talk about AMR, it's a more general category. And what we found in earlier research is that heavy metals, antimicrobials, and antibiotics have cross resistance. If you become resistant to certain, if you're bacteria, the genes that allow bacteria to be resistant to some heavy metals also allow them to be resistant to some antibiotics. And Tony Abou-Fayad is the expert on this, not me. So antimicrobial resistance is a major issue because, as some of you or most of you probably know, if we lose, and we are gradually losing the ability to treat bacterial infections, that means basically we lose 100 years of medical progress. We return back to the 1930s. We can't do surgery anymore. We can't, well, we can, but you're likely to die of an infection, so you'll only do it if you're desperate. Chemotherapy, same thing. Basically, most life-saving medical treatments that we take for granted will become too risky because of their infectious complications. So this is a major public health issue recognized by WHO, et cetera, we'll learn more in this meeting about the link between conflict and antimicrobial resistance, which is still poorly understood, but growing concern, particularly in current times with these just heartbreaking conflicts or wars, full-scale wars going on. And we'll also learn about why it appears that we're not able to develop antibiotics that work against AMR organisms that are resistant to antimicrobials. So I'm running out of time, so I'll stop there, and I'll introduce our first panel. So who's chairing the panel? Is it me?
If you want, or I can.
So why don't I pass it over to you, and then I'll chair the next panel. Maybe you can introduce our speakers.
Thank you very much.
And but maybe are there any questions? Thank you, everyone.
Thank you very much, Mingyuan. So the first panel is so exciting to have. We're going to go from China through Lebanon into India and really go from manufacturing all the way to phages. So really fascinating panel. We're thrilled to have the three of you here. Anthony's here, but thrilled to have you here, Victor, and thrilled to have you online, Mingyuan. Mingyuan's work on China is really the only work that's been done on this. So I find myself citing Mingyuan whenever I want to write about China. It's been a pleasure to get to know Mingyuan over the last few years of the project. She's also been working on a really interesting project looking at trajectories of antibiotics out of China through India and into Africa. So I'll hand over to you. Thank you so much, Mingyuan, for joining us.
Thank you so much for inviting me and for your introduction. And hi, everyone. Yeah, my name is Mingyuan Zhang. I'm just trying to share my slides. Can you see?
Yes.
Should I play it or it's okay like that?
I think you should play it because we see the sidebar. Yeah.
Okay.
Perfect.
Now it's better, right?
Yeah.
Okay, great. Yeah, so I am a researcher at University of Oslo at the Department of Community Medicine and Global Health. And just a little bit about myself. I'm trained as a sociocultural anthropologist in Canada. And my PhD project had nothing to do with pharmaceuticals. My PhD was mainly done at a sugar plantation managed by a Chinese state-owned corporation in northern Madagascar, and some other contexts of Chinese-Malagasy encounters but then after I finished my PhD, I came to this project as a postdoc. From Asia to Africa, antibiotic trajectories across the Indian Ocean. This is based at University of Oslo. And this project, like Nadia just said, was trying to trace antibiotics from its manufacturing in China to maybe formulation in India and also to consumption in Africa. And in this particular case, we are looking at Tanzania. But my role in this project is mainly looking at China to look at the Chinese manufacturing of antibiotic and APIs. So that's how I came to do research about antibiotics from China. And then most recently, I'm fortunate enough to combine these PhD interests and postdoc interests. So I'm looking at antibiotic trajectory circulation in Madagascar. But today I'm going to only, I think I'm only going to talk about the part about China. Yeah, because I have 20 minutes. Yeah, so this project, a bit of the background of this project, of course, is based on this so-called new pharmaceutical geographies from the 1990s. And China and India are really playing a big role in this new pharmaceutical geographies because now we have realized that China and India are the two major producers of antibiotics and especially key antibiotic active pharmaceutical ingredients. But both countries have a long history of national production of antibiotics, and I'll get that part of China in a bit. So today I'm just going to kind of generally introduce what I've done in this project since 2021, some of the results. And I have to admit that I came into the project at a really bad period of time because the postdoc lasted 2 years, and that was the same time as China was in strict lockdown because of COVID. So as an anthropologist coming into this project, I, of course, was thinking I would go to China and talk to people and find out more information, but that was simply impossible during this period. So a lot of the results of what I'm going to present today is really results of some alternative ways of gathering information and doing research, trying to find out whatever I can through different sources. And then that came to the combined results of these things. So first, a colleague and I, we looked at the history of Chinese antibiotic production. And really, if you want to tell the story of Chinese antibiotic production, you cannot avoid the story of North China Pharmaceutical Company, or the other name is FINPHARM. This company was the first and the biggest and the oldest company that was built after the Communist Party took office in China in 1949. And the main production of this company was antibiotics, and has been antibiotics even till today. It is still one of the biggest producers of antibiotics and APIs. It is, I think, it produces, in China, it is the biggest, and it really becomes a producer for many companies that produces antibiotics. So this pharmaceutical company, North China, it was built, it started in 1953, and it started its production in 1958. And it was really a Soviet Union style factory, because it was built in collaboration with Soviet Union, with Soviet Union aid, because at that point China was cut off from the Western world. So so we can see the buildings are Soviet Union style, and the first these are from the internet, so they're a little bit blurry. So the first picture is an older picture, and the second one is more recent, and the building is still there. It's kind of a landmark of the city, which is in northern China, a little bit south of Beijing. And Dr. Ernest Chain visited this factory in the 1960s, secretly, because it was a bit complicated because of the politics. But he came and he wrote a report, which is archived in Wellcome Trust. And his impression of it is that, you know, Chinese people, Chinese scientists were nice, but the facility is Soviet Union style, so it's not viable economically in the long term. So that was his conclusion. So as you can see, this factory is just one example of an array of factories that were built right after the establishment of the communist regime. And most of the factories had some background with the military, because the oldest factories, they were built with military support, and antibiotic production was a matter of national pride. And the fact that the Chinese in the 1950s and 1960s were able to produce some of the antibiotics on its own, it was a matter of national pride. And the products of these factories, sometimes I think they were used to help other communist countries at that time. So that was a bit of the history. And then after so many years of reform and changes, the factory still exists, and it evolved into the North Pharma company, that it's still now the biggest one that produces antibiotic APIs. So that is some of the history. And then the other ways of doing some of the research, because doing things in person in China was impossible. So I was trying to look for ways to find things. So one of the things I found, which was great, was there were many pharmaceutical trade fairs. And during this period, a lot of them were online, which was also great because I could just attend sitting in front of a computer. And on these trade fairs, not only there are exhibitors, so you can actually see who are the producers of what. You can find the catalog, like what we can see on the photo on the left. The first one is the company that I just talked about, North Pharma. And then there's also this one that down there called Qilu, which I remember Nadia and I were talking about it because it had an explosion and it just created supply shortage. So actually I was able to find out a lot of information about who produces what. And then on these trade fairs, you can also see how antibiotic APIs are sold or exhibited or marketed, or the lack of marketing. So as we can see on the right side, these are, a lot of them are, I think all of them are antibiotic APIs. Like the first, for example, is penicillin G potassium, and the second is 6-APA. Yeah. So usually there is not much marketing done about these products. They're just put on the website like this in barrows or in the, the chemical structure, picture of the chemical structure. And there's no price. So you can't really know how much they cost unless you send an official inquiry. But in a way, you could see that what is being sold there and what is being produced by these Chinese companies. And another really good thing about these trade fairs, there are a lot of events included, including talks and panel discussions. And it's really like what we're doing now, but it's just like people in the pharmaceutical industry gather and come to talk about interesting topics that, that is relevant. So I was able to attend to some of these talks. For example, this one was particularly interesting. So I found his talk. I watched it live. This guy, he is a CEO of an Indian antibiotic company, and he's very nationalistic. And he came to the talk wearing this, like what they call is a Nehru jacket. It's very pro-India nationalism. And he came and he quotes the prime minister, and he said, he really complained and decried about Chinese competition about antibiotic APIs. And so from these talks and events, I was able to kind of get into a glimpse of, what's going on in the pharmaceutical industry, and people from Europe and North America, from India and China, they all come together and talk. And it's actually quite open discussion, because I don't know, a lot of the discussions were in English, even though some of the events were held in China, but it was quite international, and it has a lot of information. So that is an interesting experience for me. And another source that I was looking at was actually, I actually went to Geneva 2 times. I was trying to look at the WTO library and archives to see if there is any files or information about, the complaints that's made between, or the disputes between Indian pharmaceutical companies and Chinese pharmaceutical companies. But it was also unfortunate that the time I arrived in Geneva, there was Omicron outbreak, and the WTO library closed without any notification. But I ended up looking around and found this from the WTO's files. I actually did this form manually after looking at 360 files of from the WTO, and I was making a very big spreadsheet to see what kind of things that has been brought into dispute. And I figured out these from 2000 and 2021, these are the types of antibiotic APIs that has been brought into attention. They've never been actually brought to the WTO, but the Indian government investigated them And because Indian companies were complaining that Chinese companies were dumping these antibiotic APIs to the Indian market, which has caused harm to the domestic industry in India. So the good thing about this is that after I found that these cases were made, I was able to get into the Indian government website, and the investigation reports of all of these cases are there and they are publicly available. So I downloaded some of them and read some of them. And it has very interesting information about, you know, who is involved and how different actors have different opinions about what the Chinese companies are doing, et cetera, et cetera. So because of all of these ways of doing research from the periphery, I would say, so I was able to write this article with a colleague of mine in my project. It's called Antibiotics Dumped, Negotiating Pharmaceutical Identities, Properties, and Interests in China-India Trade Dispute. Yeah, so in this article, we basically argue based on all of the things that we found on the, you know, from the WTO, from the Indian government, and from media reports, and from the trade fair, that there are a lot of complaints from Indian companies, but the relationship between Chinese companies and Indian companies is much more complicated than just competitors. Because in these reports, you could see that some Chinese companies are actually subsidiaries of Indian companies. So when we talk about the national identity of a drug, of an antibiotic product, even if it says it's made in India, there might be some Chinese components. So it's actually more complicated than that. And we also found that because of the structure of the international trade, it has to follow certain procedure and certain terms to establish if the case is valid. Because of that framework, that these actors, the Indian and Chinese actors, were able to negotiate over things that seems to be objective. You know, what is a chemical and what chemicals are the same with other chemicals, but they were able to argue about that based on the framework of the international trade. So we also found from this article that we wrote that a lot of the Indian actors, they have different standpoints and different points of view. So some of them definitely argue that the Chinese are competing with them and the Chinese are dumping, and the Indian government should put a tax on Chinese products, but some don't, because a lot of them actually heavily rely on Chinese imports. And if there is a tax, then they actually lose money. They actually don't make as much money. So there are many different kinds of voices within the Indian pharmaceutical industry as well. The other work that I was doing from this project was about shortage. So the first article on the left, it was more about the pharmaceutical trade fair, and I was writing a little bit about the challenges in the methodology, and how, you know, attending pharmaceutical trade fairs has allowed me to kind of get some information. And I was also able to interview one American fermentation expert who has been working in China since the late 1970s in antibiotic factory. And he was involved in the international knowledge transfer between America and China. And he is a very interesting case himself, because he is a student of Dr. Ernest Chain, and he was heavily influenced by him. And he went to China with this spirit of scientific internationalism, and he was thinking that he was promoting that China should produce more antibiotics as a fermentation expert. But then he told me after 2004 there has been no fermentation capacity in the US, and all of the fermentation facilities for antibiotics has been moved mostly to China and some to India. And he himself, his life also reflects this. He had to, in his 60s, go back to school to study so that he could work in a pharmacy in the US. And because his expertise in fermentation just is, there's no use anymore in the US. And finally, I wrote this article on the right side with a colleague who is a medical historian. So we were looking at the shortage issue from a historical point of view, more from a historical point of view. And we realized that even though now we are talking a lot about shortages, especially after the COVID pandemic, the problem of shortage existed way before this. And you can find cases of shortage reporting from the 1970s, 1980s. But we were able to conclude that the problem always existed, and the pharmaceutical industry of antibiotic manufacturing kind of always relied on few suppliers of APIs. But the only difference that now it has become a problem or issue of national security or an issue of geopolitical challenge is because now the major suppliers are in China, and they were outsourced by Western pharmaceutical companies from the 1980s and 1990s. So that's why we argued that this is an old issue, and the complexity of supply chain is always there, has always been there. But now it has become more of a global international issue because China and India and these Asian countries are involved. I think 20 minutes is up. Yeah. And then now these are just pictures of my most recent trip to Madagascar in May and June. So I was following antibiotics that circulating outside of the health facilities. And I could say, based on what I see, all of these amoxicillins, ampicillins, they are all made in China. And thank you very much.
Thank you. I should have said at the beginning that we'll take the 3 presentations and then we'll come to questions and discussion at the end. So thank you so much, and I'll pass over now to Anthony, and we move to Lebanon.
Hello everybody. Let me just share the presentation through here. I'm going to be talking about biorepositories. It's going to be a bit of a step removed, and that's kind of the point of the presentation that I want to make. So for the last 6 years, in some way or another, I've been working on microbiological laboratories and biorepositories and trying to take a supply chain view of them, and essentially really paying very close attention to how pathogen samples move across countries and from one country to another, how they flow, and also especially the politics of these flows of pathogen samples and where do they accumulate, and that's where biorepositories come in as places of accumulation. The reason why I bring this up within this sort of frame of antibiotics is I'm hoping that this presentation is in a way one also about thinking about the infrastructures for antibiotic manufacturing and production, and then in terms of the infrastructures that aren't so visible or aren't so directly visible in that process. Pathogen samples have several uses when it comes to developing medical, what are sometimes called medical countermeasures for antimicrobial resistance, and these can include antibiotics and the antibiotic pipeline, but also medical technologies. And all of this development and all of this research and development usually requires access to a vast amount of pathogen samples, either for testing of efficacy of new medications or for testing the efficacy of different kinds of detection technologies or antimicrobial detection technologies. Biorepositories are a key part of that infrastructure, and the kind of story that I'm going to be telling today, going from kind of the global view to how biorepositories function in Lebanon, is intended to just help us think about how this accumulation of pathogen samples works and how it may impact antibiotic development or antibiotic crises or shortages, in terms of thinking of it as an infrastructure. And that infrastructure's condition what is possible and not possible. So I'm going to go through just sort of the background of the research that's led me to this point. And I'll start with kind of the global view of some international law and international regulations around pathogen and benefit sharing, but then also my ethnographic research in Lebanon and my new project, or the one I'd like to do next, which is on diasporic microbiology. So I entered this research a few years ago, working on this topic called pathogen access and benefit sharing, where it turns out that there are bodies of international law that exist to govern how pathogens move across countries and allow or not allow. And there's a loose governance of it, but it comes from a place. Well, the story starts with the Convention of Biological Diversity in 1993, which is a convention ratified by maybe 200 countries and exists to protect ecological biodiversity, but also to protect against biopiracy, to protect what are called genetic resources. For the most part, this, and it protects genetic resources, also pharmaceutical innovation or pharmaceutical development has been a big part of the story, because this convention was very much created to go against practices of taking traditional knowledge from indigenous societies, or also taking plants and medicinal plants and creating pharmaceuticals out of them. And for a large part, the convention has been applied to plant resources. But within the text of the convention, microbes are also considered to be genetic resources. And what that means is that the convention states that a country's pathogens and a country's microbes are the property of that country, and that a country should be able to decide who to share these pathogens with and who not to, and also to receive benefits from the sharing of these pathogens when they're used to develop medical technologies or to develop antibiotics. This work, this emphasis on pathogens is fairly new, and it really crystallized in 2010, which is with this protocol called the Nagoya Protocol, which set down some new mechanisms for enforcing the convention. And essentially the mechanisms, some of the mechanisms are called prior informed consent. So one cannot go and take pathogen samples or microbes from a country without accessing a government body and receiving informed consent from that government body for taking them. And the second part being mutually agreed terms, that there needs to be a discussion and agreement over what kind of benefits, when a country exports, when people take pathogen samples from a country, that there needs to be mutually agreed terms around what kind of benefits flow back to that country. In parallel to this world, this kind of ecological, environmental law world, there's another world of global health law, which has an entirely different approach to microbes. Rather than seeing microbes as genetic resources, here microbes are pathogens. It comes from a long history of eradication, maybe going back to smallpox eradication, where a different sensibility towards the microbial world develops. But it also had advances in terms of reconciling this issue of pathogen movement with the developments in environmental law through the pandemic influenza preparedness framework, which very much listed, developed a mechanism called standardized material transfer agreements, meaning that it set down certain benefits for when a country shares pandemic influenza viruses, including the flow back of vaccines. So in case of a global pandemic, this framework, the PIP framework, mandates to a certain extent that countries who share virus samples that participate in creating a vaccine get the benefit of access to that vaccine in one way or the other. I bring all of this up to just sort of point to kind of international law regulations around the global movements of pathogens. And like I said before, this is loosely governed. So it's a lot of this is still in the works and still under discussion, part of heated debate. But I point this out because it just goes to show that these international regulations exist, because this has been a fraught topic. That there's been a history of inequitable and unfair practices when it comes to pathogen movements, which resulted in the creation and the enforcement and the discussions that are currently taking place around this, around what's called pathogen access and benefit sharing. This is a graph that we developed back in the day from the influenza virus traceability mechanism, which is the only kind of global mechanism that traces how samples move, in this case, virus samples. And the graph shows how different strains of viruses flow across different countries. And to a large extent, gives this kind of view of certain countries like the United States and the United Kingdom being the senders, so the senders are over here, and the receivers are over there. So we kind of get two maybe key takeaways from this graph. One, that these virus samples flow from developed countries to developed countries internally. And two, that there is a very significant flow from government research institutions, as the ones that receive these virus samples, to a whole host of non-governmental institutions. And in this case, small and medium-sized enterprises and multinational pharmaceutical companies. So all of this to say is that there is a corporate pharmaceutical, multinational and small, medium-sized, these samples are accessible to companies and they're used to that extent. But going to this, what this graph also gives us, I think, is a partial view, because it gives us this idea that virus sample sharing is taking place between developed countries. This is where the ethnography comes in, because my ethnography, which takes place in Lebanon, to a certain extent tells a different story of biorepository building and accumulation of pathogens. I'm just waiting for it to update. This is the title of my thesis. And my thesis took place in 2021 in Lebanon during a financial crisis, one that the World Bank has called one of the worst financial crises over the last 150 years. My thesis work took place in different areas, some in hospitals, a lot on pharmaceutical shortages, and a big part of it in microbiology laboratories. I'm going to be focusing on this part today, the biorepository building. And this is essentially, waiting to update. This is kind of the map that informed my ethnography with biorepositories. This is a network analysis of the microbiologists who are publishing in Lebanon on Acinetobacter baumannii and resistant Acinetobacter baumannii. And it kind of just led me to where the scientific centers in Lebanon were for this kind of research. And by extension, where the microbiology laboratories that were producing this kind of research operate. And what's striking kind of off the bat is that only this small part here, which is on the upper right corner, the Lebanese University one, is the only one that operates from a governmental institution, whereas all other laboratories operate from private university hospitals. But in my thesis, I traced back this kind of the story, or the historical story, of this privatized microbiology that existed the moment I stepped into Lebanon to do my field work. And here I'm sharing this design that I really like from 1948, which is at the dawn of the Lebanese Republic, by a physician called Dr. Al-Balmati, who drew up at the time a design for a public healthcare system for Lebanon. And he kind of, he set it up in this archway design that's very much in the tradition of social medicine, with social services and medical services combined. And he gave the health authority, or the public health authority, or the government health authority, a central role within this mechanism. The reason why I share this is because, and what makes it very interesting, is that this is a kind of an imagination from the late 40s and early 50s on what Lebanon's healthcare system would look like in the future. But when I arrived to Lebanon, it was an entirely different view of a very small public sector. And an absence, and this was kind of the key motivating finding, that Lebanon does not operate a central public health laboratory. So we do not have a central governmental laboratory. My fieldwork then ended up taking place for the most part in private microbiology laboratories. And this is where I kind of developed this view of absent infrastructures, sort of arguing or speaking about how the absence of the central public health laboratory, the absence of this national level governmental office, conditioned a situation where private laboratories, for the most part, the jobs of a national public health laboratory were parcelized and distributed broadly, again, to a wide array of private university laboratories who took on different tasks managed by the Ministry of Health. And this is very much what I documented and saw during my fieldwork. I bring this up to say that when it comes to what Lebanon shows us differently than the global view that I have given before, where a lot of the pathogen accumulation was happening in government research institutions. This case from a country like Lebanon disturbs the assumptions that we begin to have about how microbial accumulation happens, which I think is very much an assumption of having thought for a long time about biorepository building from a developed world, global north perspective. The case of Lebanon is showing us how countries in the global south might do things differently, and the way we might think biorepository building and pathogen movement works on the global stage doesn't map into the experiences of smaller countries like Lebanon. And here, one of the key kind of areas that I started focusing on in my thesis and in my work has been historic microbial isolates. A lot of my work was about trying to find historic isolates and historic pathogens in Lebanon, and this was met with a lot of futility, except partly because of the absence of the Central Public Health Laboratory, which meant that there was no central biobank through which Lebanon's historic isolates could be collected. But the other part of it is that historically and across time, Lebanon's microbial isolates or pathogens have routinely been sent to institutions abroad. And this takes me back to this map that I started with, where you'll notice that a lot of these microbiology laboratories that I focused on are also attached to international collaborators, from Italian collaborators to French collaborators to collaborators in the United States and Canada. Meaning that when I was asking about historic isolates in Lebanon, I would often be told that you're more likely to find Lebanon's microbes in biorepositories in Marseille, France, in the CDC in the United States, rather than in Lebanon itself. So I bring this all up to go back to this idea of how infrastructures condition what is possible and not possible when thinking about global supply chains and what this means for the antibiotic industry. This kind of centralization of pathogen samples and their accumulation in specific places in the world means that only specific and few people can work on developing or doing the necessary research and development for the antibiotic pipelines and for the medical technologies. A lot of the microbiologists that I work with are interested in building local capacities and in opening up the possibility for this kind of research to be happening elsewhere. But to this point, I'm going to just move quickly to the next project I want to do, which is that a lot of the Lebanese microbiologists I've worked with have moved their laboratories, or I've heard about, have moved their laboratories abroad over the last few years as a result of the financial crisis. So the next leg of this work is going to be looking into this idea of diasporic microbes and diasporic microbiology. So these Lebanese microbiologists, going from being in microbiology labs in Lebanon to being in microbiology labs that contain samples from Lebanon, and where Lebanese microbiologists are now situated abroad, and starting to think about telling a transnational story of microbiology and biorepository building. But what I'd like to discuss today and to think about with everybody is to what extent biorepositories might be a blind spot in the global antibiotic production and manufacturing supply chains and pipelines, and to what extent this might be something an under-investigated infrastructure and part of the bottlenecks that we're seeing today. Thank you.
Thank you very much, Anthony. And you give us a challenge as well to think about for the concluding remarks at the end of the day. Victor, maybe hand over to you and move to India. Up to you.
Does it work? Can everyone see? Yeah, right behind you. Just get my notes in. So I've got quite a bit of images in here rather than text, just to try and think about it. So first, I'm Victor Secco. Thank you for having me here. I am a social anthropologist. I'm doing research at the University of Venice. We have a project called HealthX Cross, which is, I've put a little logo here. If you search it online, it can come up. I'm not going to talk about it too much. I am looking at microbiome towards more environmental science, planetary science. There is antibiotic resistance inside of it, but it's not the main focus of our project at the moment. Instead, what I want to talk about is the research that I've done for my PhD. So this is what I did when I worked in India, and mostly with phages. And then I did at the University of Manchester, so I've put it up there as well, the University of Manchester. And then just as a little, well, just as an information, I guess, similar to Mingyuan, I did my fieldwork during COVID-19, so that has impacted a bit of my possibilities of doing part of this research. I was in India when it started, so I was able to access it for a few months, and then afterwards things started to change, so then I had to move a bit. So as part of this research, that of course this was done in 2019 and 20, so some things have already changed as well, and I've put a couple of things that have changed. And then perhaps, well, I can't see people online, but I leave a question that I thought about asking when I get more people. Have you heard about bacteriophages? Maybe how many people here have heard about bacteriophages before? Some people, yes. I get quite surprised, mainly in the clinical, perhaps, environment, when I meet doctors, GP or someone that you might mention, because I do that research, and sometimes it comes in conversation, and of course people know what are bacteriophages in the clinical practice, but they are mostly not aware that bacteriophages can be used into phage therapy, or there is anything such as a therapy that is a different kind of an alternative to antibiotics. And so I will to phages. So if you don't know what are phages, I'll just go very quickly on what they do and how they've been working. Phages are viruses that infect bacteria, just like we have viruses, other organisms such as bacteria have viruses. So they can integrate in the bacteria's genome, and then they can either hijack, they can either stay there, or they can hijack, and this is the cycle that I'm showing here, the lytic cycle, where they can hijack the metabolism of the bacteria to reproduce themselves, and in the end end up breaking the bacteria cell, and killing the host. But phages are mostly quite narrow spectrum in antibiotic language, are quite narrow spectrum in a sense. So they tend to infect only one species or strains of the bacteria that they are kind of adapted to prey on, in this kind of predator-prey relationship that they have. And because of this relationship of predation with bacteria, phages have been, since their discovery in 1915-17, proposed as a treatment. People noticed that they could get rid of bacteria with these viruses, and said that maybe you could do a phage therapy, you know? But then, what happened to this phage therapy if we knew about it even before the discovery of antibiotics? Now, this is 1915-17, before 21, 22, I think, penicillin, isn't it? And so the question, I go back a little bit on the history of this discovery, just so that we try to make sense of what happened in that history. So just to start, and I won't talk too much about it, but my research in India was also related to this prehistory of phage discovery in India. So in 1896, a British chemist goes into the Ganges in India, and still British India, and after hearing the stories that the people were using the waters of the Ganges as a sort of a cure and sort of a curative, I think he decides to do experiments with the waters of the Ganges, and finds out that if you put the cholera bacterium in this water, it just dies in the raw water of the river. Instead, if you boil, filter, or use well water or other sorts of waters, the cholera could stay longer. So he doesn't really understand what is that, and there was no idea what were viruses at that point. But it is understood as a kind of a prehistory of bacteriophage, and India plays, and in my PhD I use a lot, India plays a lot with this idea that it was already known in a certain sense, and it comes in a sense, in a sense from also the local knowledge of people in India. But it is in 1915 and 17 that the phages are actually isolated or discovered in that sense by two different persons. One of them, William Ford in the UK in 15, and Felix Leclerc in France in 1917. And the hell, I leave the picture of the hell because he becomes quite a big figure in this, and the hell is the scientist that says these are viruses, because there wasn't an agreement on what were this. They could isolate, and they figured out that there was something killing this bacteria, and they supposed that these were agents of some sort. But viruses were not, at that time you couldn't see them, there wasn't electron microscopy yet invented, and so they couldn't make sure that these were viruses yet. But there was one of the persons who said these are viruses and we can make phage therapy out of them. He was one of the first proponents, I think, of phage therapy at the time, and he then starts proposing and creating these clinical trials at the time. So the first clinical trial that they do is between 1927 and 1937 in India, actually, which is called the Bacteriophage Inquiry, and it lasts almost 10 years, more or less, and they do it with the help of India. And of course, what happened in there is, and this is the historiography, this is the historians talking more about it, but from what I understood is you had some good conclusions of the study, but data wasn't very good. They didn't have a good understanding of viral biology. It would work in vitro, but it wouldn't work so well in vivo in the people. And of course, there was also political turmoil because this was just pre-independence India, and there wasn't a huge collaboration with the, I guess, local population, questions of applying these things into people. I think most of it was done in Calcutta. And so his history is quite interesting for bacteriophage history, because he moves in between France, India, and then he ends up in Yale at some point, not for many years. And from Yale in 1934, he moves into Georgia, in what was then the USSR, the Soviet Union, to set up the Center of Studies of Bacteriophages in there with the help of another colleague of him from the Foscari Institute in France. And this is the beginning, and this still is the biggest center of bacteriophage nowadays in Georgia, the Eliava Center, which I'll show in a little bit. But this is not very well seen in his movements. Apparently, some of the historians of phage tell that he has a particular personality that is one of the factors that helped bacteriophage not to, the phage therapy not to develop as it could. Also, because of his political affiliations moving back and forth, I think he dedicated one of his books quite controversially to Stalin, although then Stalin kicked them out of the USSR afterwards. Stalin killed George Eliava, which was his colleague in Georgia, and kind of tried to erase that idea, but the USSR kept the center of phage therapy in Georgia and continued on developing that. And then another element that kind of helped a bit disappearing with or fading of phage therapy in the Euro-American context is, of course, the discovery of antibiotics and the industrial production and the use during the war mainly, and also during the Second World War. Antibiotics become a bit more this saving, this magic bullet that thanks to penicillin will come home, this kind of magic bullet that you can use. It's industrial, you can make it in large scale, it's broad spectrum, some of it, so you can kill different things at the same time. And it kind of builds a bit of this divide in between the Euro-American, Western Europe using antibiotics, whereas Russia did invest more and use bacteriophages during the war. And although there was a bit of sharing, I was reading recently, they did try sharing a bit of these technologies. Russia had the same, or the Soviet Union at the time, had the same kind of, okay, this is a different technology coming from them, we're not going to use it, we're quite used to using bacteriophages, let's continue with phages. So then this is a bit of the history that becomes, this is the George Eliava Institute in there, and then of course Russian, Soviet, I think this might be still Russian, commercial, like you can get in pharmacies and you can get this kind of little preparations that they have. These are from Georgia and these are from Russia. And to this day, a few of the places where you can find bacteriophages in pharmacies are Russia, Georgia, and Poland, for example, are places that you can find this kind of more large-scale producing of phages. This is an image of during maybe before the war, but large-scale production fermentation of water in the Soviet Union. And so this became understood in certain contexts after the war, mainly, and with the stronger Cold War period, in the Iron Wall, as a communist technology. People, of course, had heard about phages, and it kept on moving between here and there, but in the US, mainly, it became attached to this idea of this communist technology, bacteriophages that were not really, also, I suppose, in a different context, in the capitalist context, weren't seen also as a sustainable way of producing medicine. And then the rest of the story is a bit of what we know. We start to use antibiotics. Antibiotics take over of the world and become, in a certain sense, what other authors have said, an antibiotic infrastructure for everything that we use in the medical practices. I think you're saying for chemotherapy, for surgeries, for everything that we use, it kind of creates this foundation for modern medicine. And then you have, of course, certain organisms that become more and more problematic. And this is one of the cases that I studied in India. So from early 2000s, because of antibiotic resistance, because of antimicrobial resistance, so a bit of interest in phages started coming up again in the early 2000s. So one of the persons that I worked with in the University of Varanasi in India was a professor of microbiology who was exactly his research was to go in the Ganges, look for bacteria of this group, of the escape group, which are the pathogens most problematic for antibiotic resistance, as far as he was telling me, and finding the phages that could infect this. So, because of course, to isolate phages still from the environment, you look for the bacteria, then you look for the phage in the bacteria in it. So the project was to look for the bacteria and the phage, and he found all of them in the Ganges River, and more, typhoid and other kinds of things. And of course, high levels of resistance, you get a lot of sewage going into this river, you got a lot of people using the river for bathing daily. And these are parts of the PhD. I won't go too much in detail, but I looked a bit more into this relationship between rituals and the microbiology of the river in a sense. And then what I wanted to go a bit more on the financialisation of phages in India is the part of the work that I worked with this biotech company in South India. And these are just some images of conferences that I attended with the little phages that they make in the Indian style in Delhi. And this huge promotion that I felt in this conference and in India in general, working with microbiologists and these companies, pushing this idea of phages for cure, not trying to make this breakthrough of phages to outside. So I worked with this company in south of India, in Bangalore, where they are trying, and I think they are perhaps the first company, biotech, not government, not a university institution that has public funding, but a biotechnology institution that tries to make phage therapy, tries to get them out, phage products out, and commercialize them. This is a person who comes out of California, early biotech industry. He's Indian and he decides to come back to India and set up this this biotech in early 2000s. And of course they run to the problem of phages can't be patented. They are viruses, and as a natural occurring entity, you can't patent phages. And so it's quite tricky for them to actually make any money out of phages as a company. And so this was one of the first things that I noticed when I was there. They don't call themselves a phage therapy company or anything. It's phage-derived solutions. So they try their ways around phages to make them profitable in a sense. So one of the ways that they started doing it was, for example, they isolated a phage that was very potent and useful to treat MRSA, no? So multi-resistant Staphylococcus aureus, which is has become quite complicated in the clinical practice, in surgery, and it's a problem that perhaps sometimes the person doing the treatment, no, the clinicians, the doctors, transmit that to the patients in that sense. So they isolated the enzyme in the phage, in the tail of the phage that breaks the cell of the wall cell of the bacteria, and then they could patent this process of isolating and using this enzyme itself. So it's not a phage in itself, but they use the enzyme of the phage to create. And while I was there, and I think it is still the case, they're in the final steps of a clinical trial for using a nasal gel to be used as prophylaxis in the medical staff before doing any sort of surgeries or engagements. This was one of the ways that they were trying to get around the problem of how to make phages profitable in that sense. They were already 20 years in the research. They started in 2000, I think, and of course 20 years of investment without any sort of return for them at that point. And I think, if I have understood quite well, it's not out yet, so there's still 20, let's say 25 almost years in without a return. So a bit of what I'm pointing here is that it's quite tricky to make phages work for the way that the pharmaceutical industries are financed and are taught with in the system, where you're based on patents and you're based on making it. You could, of course, and George Eliava Institute that I showed provide phage treatments, and of course you charge for the treatment and you charge for all laboratory things, but these are different from making a product that you can put on the pharmacies and sell it over the counter and make money out of the royalties or out of the patents of it, in a sense. Because what can be done, on the other hand, because of the narrow spectrum of phages, that you could produce them in large scale, you could do certain cocktails, which I think is what the Eliava Center does, what happens in certain places like Russia. But the regulation in a lot of countries is very tricky in relation to that, because phages end up in it, and this comes more really in Europe. It's not so much what I have done, but other people have been talking about it, writing, I'll show a couple of names after. In Europe, phages, for example, were, I think it just started changing, I'll show you as well the regulation, but phages were understood as a drug. And so as a drug, and they would basically fit in the regulations for antibiotics, if you would put it like that, as a pharmaceutical treatment. And in that sense, it was very tricky to do not just clinical trials, but also to control dosage or any form of this production of these cocktails that you would have to do. And what happens a lot of the time, these cocktails might not be useful, so the phage Eliava Institute, you go there, they take your samples, they work with the phages to create another phage that might work for your specific case. So it's a very personalized kind of medicine. And then just to finish, is this idea, I think, I thought about this idea of the communist technology and the communist virus might still be an interesting way of thinking about phages, because perhaps of their own biology, perhaps of the way that they fit in the systems that we've created, phages might still sound like a communist technology. It would perhaps work in a lot of ways, but it doesn't work inside the systems that we have, the pharmaceutical, economical cycles that we have. Phages are not attractive as a product, as a product. No? So you do have, these are some of the books that have come out lately talking about, not all about phages, most of them are talking about phages, and this idea of the friendly and good viruses. You know. And this is just an image. If you search for communist virus, they actually come up with a phage virus. So I thought that was very interesting. But thank you very much. It's just some questions were kind of out of it.
Thank you very much. So now we've got half an hour to spend discussing questions, sharing. Are there any questions in the chat? And please, people online, just speak, or we'll monitor the chat as well if you want to write. If you want to write into it.
So I'm trying to. So first of all, I think just an observation across the three papers, and I'm sort of practicing for my concluding remarks. So one is the sort of clearly transnational infrastructure has emerged here as a common theme. And so there's something about how things circulate and how the circulation is shaped by historical regimes, you know, communist viruses, regulatory disputes, post-colonial developments, and so on and so forth. So I had a question about the Chinese story, and I'm wondering about. I think it's a question to both of you, which is that there is this. There's clearly a socialist. No, there are many. There are several socialist. What's the word? Not policies, something broader. But there's a socialist visions about investment in antibiotics in Maoist China and phages in the USSR. So I'm just curious to hear from both of you how would you characterize this antibiotics, or I'll include phages in that, sort of antimicrobials as part of a socialist project. So maybe Mingyuan first.
Yeah. So I would say at the very beginning, when it was established in the 1950s, all of the pharmas, they were actually called socialist big pharma. So there is definitely a general socialist vision behind all of this. And also there was this collaboration between different pharmaceutical factories at that time. It was not, the factory, first of all, the factory was not built to make economic interest. The factories were built to collaborate and to share. So at that time, the one that I shared was the biggest, but there were others that were built, and they kind of shared. For example, some factories would produce some types, and then other factories would produce other types, and then they have to kind of collaborate, and they collectively supply the Chinese need. Or if they have extra, they will supply other communist countries. But there was this spirit of collaboration and sharing at that time, in the 50s and 60s, and even in the 70s. And another thing I wanted to say is, even though, of course, on the surface, it's socialist, the big pharmas, but it was not so completely cut off from the Western science and international knowledge transfer. I was surprised to find that person who actually was working in China near the end of the Cultural Revolution. And he's an American, and that just means that the Chinese pharmas back then, they knew they needed something more than the Soviet Union technology and expertise. They've always known that. And the very first penicillin that were brought to China were from the United States. And that was brought to China before the Second World War. So that was when the US and China are so friends. So the American and the Western influence has always been there. And there was a period that in the 60s and 70s that China was also breaking off from the Soviet Union as well. So there was a struggle to gain national self-reliance without any help. But they also knew that it was not possible. But I would say at that period, this was definitely part of the socialist state building process. And the achievement of pharmaceutical manufacturing was something that it was part of the socialist vision. But then after the reform in the 1980s and 90s, then the nature of all of these companies have changed. They've changed to, even though they might be still state-owned, which means that they receive state subsidies, but the goal is to make money, and they participate in the same international pharmaceutical market. This actually creates a problem today because people think that, well, these big companies in China are monopolizing the production, but in fact, these companies also within China, they struggle to find their own blockbuster drugs. They struggle to, keep this traditional production, but they know that this is low value creation. So they are also trying to produce other types of pharmaceuticals that will have more added value.
So maybe just to, before turning to Victor, so there is this kind of what would appear to us this utopian vision of what we would call public good, that these companies collaborate, if there's leftovers, they send it to brother or sister nations, this kind of thing. And this ends with the reforms in 80, the free market reforms, or it starts to dwindle out before?
I think the real change happened after the reform that started in 1978, because after that, the companies are starting to get private investment, and they are participating more in the free market and in the international market. But more of it also happened after China joined the WTO in the early 2000s, and also I think in the 1990s, China joined the International Agreement of Patent Protection as well, and that also did something to the Chinese pharmaceutical industry. But I think the real change happened after 1978.
So Nadia, just a thought, because you had talked about the Chinese industrial policy, it'd be interesting to trace that back. Anyway, Victor.
Yeah, thank you. I'm not a huge expert on the Soviet Union or Russian context, but maybe perhaps what I could say also in relation to phages is the history of microbiology in the USSR, in the Russian context, is slightly different from what I understand on the history, but it's a bit more ecological. They always had a bit of a stronger focus on the ecological side and perhaps more relational, and phages are quite interesting in that sense because you can source them from the environment a lot. You use a lot of sewage water because that's a good place to find phages that affect us. Sewage is a place that kind of resembles us, our insides in the outside. And so perhaps it kind of fitted well, I think, the idea of phages, although they were, as I was explaining, discovered there in France and the UK, but it kind of fitted well in the developments that were happening already in the microbiology of Russia, more than the very antibiotic perspective that perhaps took hold in Western Europe and the US afterwards, after the discovery. But I don't know much more about the history of what happened with phage production inside those contexts afterwards. I don't know how much is written about it. That there might be something, but in detail of what happened in the Iron Curtain, I'm not too sure how much is.
I have this vague recollection, so correct me if I'm wrong, that phages were, there was kind of a tradition of Soviet science and medicine, which was intentionally or not positioned against Western science. So they did, as a Western doctor, they did all kinds of crazy stuff, right? Including Lysenko, right? Lysenkoism, phages. There's a bunch of stuff that, in my mind, are all associated with kind of a retro, charming Soviet thing, which, Lysenko, there were clear ideological reasons, right? So Lysenkoism, the idea that organisms can, environmental influences on organisms can change the organism, and that can be passed on or transmitted, or the inheritance of acquired characteristics, that's the term. So obviously that fits with the Soviet idea of a new man. Phages, I'm stretching this a bit, but is there an ideological kind of convergence around this idea of environmental, right, less individualized, more communitarian? Do you think there's a kind of an ideological, have you encountered that?
I think it could, yeah, I think, I mean, it has become a communist technology. I think that is a word that comes out mainly out of the US for phages. In
the 70s and 80s.
In the 70s and 80s, it's really understood as a technology that is coming from that context.
But in a pejorative, in a negative sense?
In a negative sense,
as kind of communistic, lazy,
and
communal.
Yeah, and attached to the ideas that this is coming from the other side of the world, and we don't engage with it as much, perhaps. In the USSR, because the tricky thing is they are quite I guess you can produce them in large, and they make these cocktails, and they were using these cocktails in metal. But the Eliava Center, for example, and I suppose they might have other centers, it is quite individualized. In the other hand, these therapies. Now, what happens
There's a very amazing documentary that probably you've seen from Germany. About a patient who cannot be treated anymore in the German health system. And the accusations, it links a lot of what we've talked about for me this morning and what we're going to talk about later in financialisation, because one of the reasons for the failures of the German system in this documentary are financial pressures on the system. And this patient cannot in any way be treated by normal antibiotics anymore and goes to Georgia. And it's something like, it's about also the implementation of DRG, the DRG payment systems, which come in my paper later in the US, and impact on antibiotic markets. It starts there and it ends with the patient in Georgia being treated in that, with this very personalized treatment that saves the person's life, that's not available in Europe.
Because it's perhaps it is a bit more of a care of a community, that you would need to invest money for this kind of individual treatment that is a bit more expensive for the government, if it is done by a government or for the institution almost, than for the I mean, in a sense, it didn't fit, I think, the US way of thinking of phages in that sense.
And isn't it fascinating that they can't be patented, right?
Yeah, exactly, yes. I mean, I think they try in the US, and they keep on trying, because at some point you could patent almost anything, but it's not... recently then you do have, I just saw, I think the book from Tom Ireland, the Good Virus. He talks about really recent stuff that they've been doing, and there is a company trying to create artificial phages based on artificial intelligence designing of it, and then they can patent all of these sorts of technologies. But the virus itself, and also the question is, even if you patent a phage, it might be useful for one person, it might never be useful again, because there's also, they're in a dynamic relationship, so there is no point in identifying.
Widely commercialized.
I really like one of the concepts that you talked about, Anthony, about absent infrastructures, because I think that was present in all three presentations. And your concept of, look, things being done differently here, but then the sadness of where you're next going to go for your field work, which is going to be there again, with then this concept that came through in both Mingyuan and Victor's presentations of the pressures that then the normal financialized biopharmaceutical system then puts or takes from these local knowledge bases, local production, local, the fermentation capability really only being in China, the phage capacity not being accepted or acceptable and then your concept of the transport of the material needed already being out of the country, but now all the people as well, because the people part in the antibiotic space is incredibly important too. These absent people in the whole development space and the production space is also an incredibly important part of why we're in the mess we're in. So I really liked that.
In many ways, it's a problem of regulation. I think for me, what's coming out of all of this is also what could be possible, but is not possible for one reason or another. For reasons, but mostly for reasons that have to do with what's commercializable and what's available. In my case, the material that's needed to produce this technology is no longer available. It's available elsewhere. But I don't know if it's too much of a stretch of the imagination to think about how these alternative ways of doing things can be put forward as solutions that can work on a broader level.
Well, I think that's what's coming out in a way, that there's an imaginary here common across a kind of a socialist imaginary, which I'm not sure if that's viable as a model for market failure, right? There's a bigger narrative here from this vision of the Lebanese healthcare system, and how do you put this? To come back to infrastructure and sort of operating standards, right? This is what we require globally, because this is what we see enables all. You need that interoperability and those infrastructures to do all these things, and this is what's fragmenting now, in Lebanon, USSR, there's something left in Georgia, China, and so on. The nationalism of sort of what do we call it? Therapeutic sovereignty.
Yeah.
The sort of the national security arguments actually go against this kind of global infrastructure.
Well, it's really interesting, Mingyuan, I think in some of your recent work that you're pointing out the economic challenges that the Chinese companies are facing, where you just pointed to this kind of utopian socialism, but some of that utopian socialism is still very much present in the academic literature, talking about nationalizing production, talking about more state intervention. They don't call it like you just called it, of course. They say we've got a market failure in the world for access to antibiotics, so we need more government intervention. But you have this kind of dichotomy between these financial pressures that you've mentioned coming through in China now, and shortages that are coming through in China now, that you've mentioned in some of your work, that I think are very, they're very not known in the West, that's what's going on there. While there are calls in the West for more government, and more government.
Yeah, I think it's important to think about these Chinese pharmaceutical companies as part of the world economy rather than as anomalies of it. Because what they are doing now, not after in the 1950s and 60s, but what they are doing now is fully participating in the world economy, even if they receive government subsidies. But they have to make money. So what happens with the Chinese antibiotic manufacturing is China is not reaching its capacity limit. China can produce a lot more antibiotics and APIs, but China is producing this much, and then these traditional companies are looking for ways to make more money by switching to other kinds of more advanced, more R&D involved, more added value products. So they are also kind of, this is the product we have to do. Like one person I asked told me, I said, it's so cheap, antibiotics are so cheap, and that's why no one except the Chinese ones want to produce it. But then this person said, well, someone has to make this money because there is always going to be a need for antibiotics. So that's the problem. And the only reason that these Chinese companies are doing it is because there is this effect of volume. So because China is the only place where they can produce such large quantity to make sure that they still make money from it. With help of energy and chemicals input, that's why. And that's why the Indian companies also cannot compete, because they don't have the support from the government and the cheap input. But this might also change, because as Chinese economy is changing, so there might be changes, but as of right now, this is how it is. Yeah.
Very interesting.
It's kind of built on that, the paradox and the interoperability question. Paradox here is that the financial systems of the world speaking to each other is supposed to be making things more interoperable, and making it possible that these sort of niche technologies, what are currently niche technologies, would be able to develop as tangible solutions. But instead, everything is showing the opposite.
Yeah.
It's monopolies. And at the end of the day, it's about
Apple and IBM.
So
or Android.
The promises of global capitalism are just clearly starkly not working.
And your Indian biotech that hasn't made any money for 25 years.
Yeah, how are they managing?
I think they keep on getting funds from
California.
Yeah, I think there's still a lot of money behind that, as far as I know, and they try to connect with other pharmaceuticals. I mean, they are very well connected people in the pharmaceutical. There in Bangalore, there's a lot of other pharmaceuticals. They are somehow surviving. It's not a huge operation, but they do have a little operation there. But at least when I was there, I could see that they're clearly worried about it, you know, in a sense, like it's a big investment, and they want to get it.
Yeah, I mean, the antibiotic biotechs in the US, it's the same problem. 15 years, spend money, and then you can't make money at the end unless you get taken out. And even if you do, you get taken out below cost.
Hi, can I jump in on the conversation? Right, so there is two concepts right there that we need to address. First of all, we don't need companies to manufacture more antibiotics. What we need is new classes of antibiotics that don't have the same mode of actions as the antibiotics that do exist in the market. And this is one thing that we are badly suffering from. So I'll give you a small example. We discovered carbapenems, and then they started making these carbapenems with these carbapenemase inhibitors like ceftazidime avibactam. Now we've got vaborbactam, relibactam. All of these are being added to the arsenal of carbapenems in addition to carbapenemase inhibitors. But the problem is not there. Instead of focusing on finding carbapenemase inhibitors, we need to find new antibiotics that work in a different way. And this is one of the main issues where we face problems. And as one of these who emerged with a PhD from this topic, so my PhD topic was discovery of novel antibiotics, and half of it was funded by pharma companies when I was in the UK. And the idea was we would hire PhD students to do the R&D because they are cheaper labor. They can do the legwork at the universities, and whenever there is a hit which deserves to be taken forward, we can take it back into our pharma companies and push it forward. This was the original approach, but even that is not really applicable because the main source of antibiotics is actually bacteria or microorganisms, all in all. Because yes, we humans, we are smart, but we're not that smart. No offense to anyone. Bacteria, they do produce these antibiotics because they live in a community. In a handful of soil, there is more than 1 trillion different species of bacteria that live together. And they have to fight each other for nutrients and for food. That's why they make antibiotics that kill other bacteria while keeping themselves safe in order to survive. And what we did right there, we went into those niches with isolated bacteria that produce antibiotics, and we've taken them forward. And you can count most of the antibiotics that we use, be it carbapenems. The mother molecule is called phenamycin. It's produced by a bacterium called Streptomyces cattleya. Colistin, which is another last resort antibiotic, is also produced by bacteria. You look at the tetracycline family, it also comes from the same source, so on and so forth. I'm not going to count them all, but they all come from the environment. The problem at the moment is during the antibiotic golden era, during the 1940s until the late 70s or early 80s, the rules and the regulations of FDA and other authorizing bodies were a lot easier for antibiotics to make it into the clinic. But if you look at the rules right now, and if you would take all the antibiotics that we have and you put them back into FDA trials, they will all fail. Because it's not that easy to find an antibiotic which is not nephrotoxic. It's not that easy to find an antibiotic that does not have some sort of a side effect somewhere on the human body. So we have several problems that are all conglomerating together, all attaching each other, hampering us from discovering new antibiotics in order to find new antibiotics that could actually help us. And one of the other problems is as well phages. I'm one of those who don't believe in phages very much, and I have a scientific reason for that. And that's because, one, the resistance rate of bacteria to phages is very high. The fact that it has to be personalized medicine, quote unquote, so phages would be designable phages, and this is not easy. Third, if you go looking for natural phages, you know that it would kill this pathogen specifically, but you don't know what it would do to the rest of 1 trillion different species that live in our gut and in our microbiome. So phages could be used in certain cases as last resort, but there would be side effects that we don't understand. So the actual solution for the problem that we are in with the rise of antimicrobial resistance, yes, you are right, pharma companies are not making money because they make a lot more money with medications that are for chronic diseases. But somebody needs to put an end to this money-making machine and get them to understand that we need to unify our efforts to get to places where we can actually start treating people with new antibiotics. I'll give you a small example. When I arrived to Beirut in 2017, we used to get 1 or 2 cases of pan-drug resistant bacteria every 2 to 3 months. Today in 2024, we get 1 or 2 cases per day, and that's 1 hospital. Pan-drug resistant means resistant to all clinically utilized antibiotics. So we try to come up with combinations that could guide the physician to give them a treatment. But does it always work? The answer is no. Is the mortality getting higher? Yes, absolutely. Is it focused only on gram positives or gram negatives? The answer is no. It's all bacteria are becoming like this. And as I mentioned, bacteria are very smart because they can adapt quickly. You give them a new antibiotic and you will raise that resistance within a few weeks, because we don't have these policies that you guys have in Europe, for example. I mean, when I lived in the UK, I got sick. I was not given antibiotics until day number 7. And I found that to be, at first, I found that to be, oh, this is horrible. I mean, people can die. But it turns out, no, this is the right approach. While here, buying antibiotics off the counter, like you buy bonbons or Halls or strepsils, is the wrong approach. So we need to apply policies in countries that don't have policies. And unfortunately, these countries that don't have policies, their population percentage is a lot higher than the countries that do have policies that apply. In India, you don't have policies. In Pakistan, you don't have policies. In Egypt, you don't have policies. That's already more than 2 billion people out of 7 across the world, right? So this is already a reservoir to raise resistance. So one approach is to implement the policies across the globe. Yes. Is it doable? They have to figure something out. The policymakers, and I'm pretty sure Vin Kim is probably smiling now, and look at the other end of the problem is to discover new classes of antibiotics that kill bacteria in a new way and keep those as a last resort to be used at the right time, not with everybody's hand. You shouldn't be able to go to a pharmacy and buy meropenem or imipidine or colistin. But unfortunately, in our area of the world, this is very easily possible.
Thank you very much, Tony, for putting the real crisis on the table to close this first panel. We're going to stop then now for an hour to have lunch, and then we'll come back to your panel afterwards. I'd like to thank again all 3 panelists for taking us around the world in this fascinating way, and we'll see everybody back in an hour. In the chat, we put again the link to please give us your email if you want to join the community of practice, and we'll see you back at 1:30 Geneva time. Thank you.
2:30 Beirut.
2:30 Beirut.
Thank you.
Thank you very much.